Cells. A lion, a sunflower, a spider, or a mushroom all look different from the outside, but on the inside they all are made up of somewhat similar tiny building blocks. These building blocks are present in all living organisms and were first discovered by a British scientist, Robert Hooke, in 1665 and were named as cells. Later, with the advancement in the microscopes, it was discovered that although all living things are made of cells, but they differ in the number of cells and types of cells. In 1925, Edward Chatton, a French microbiologist, discovered that in some cells the genetic material is freely floating inside the cells, and in others it was enclosed inside a membrane. Chatton came up with the idea of classifying the cells into two types: the simple cells with freely floating genetic materials as prokaryotic cells, and the others with the genetic material enclosed inside a membrane as eukaryotic cells. Let's look at the classification of living things one more time. Earlier, the classification was based on the physical characteristics of the living things. So all the living organisms were classified into two kingdoms, the animal kingdom and the plant kingdom. Previously, fungi, for example mushrooms, were classified within the plant kingdom as they did not show active locomotion and some of them used to grow in soil. Later, scientists discovered that unlike plants, fungi cannot produce their own food through photosynthesis as they lack chlorophyll and depend on other organisms for food. So this and some other unique characteristics led scientists to classify fungi as a separate kingdom. After classifying living organisms in these three kingdoms, it was discovered that some other types of organisms are there, such as amoeba and algae, which do not show common characteristics with animals, plants, or fungi. Therefore, another kingdom, Protista, was created to adjust these living organisms. Later, with the distinction between prokaryotic and eukaryotic cells by Edward Chatton, another kingdom, Monera, was introduced, and bacteria and archaea belonged to this kingdom. If we look at the classification of cells by Edward Chatton, then bacteria and archaea, they have prokaryotic cells, and animals, plants, fungi with mold, mushrooms and yeast, and protists with algae and amoeba have eukaryotic cells. Let's talk about a cell and the cell organelles. A cell is the basic structural and functional unit of life. Cell organelles are the subcellular structures which perform a special function. Here we are talking about a typical eukaryotic cell. Every cell is surrounded by a cell membrane, which controls what goes in or out of a cell. Each cell has a jelly-like substance called cytoplasm. Nucleus is the control center of a cell. Mitochondria are the powerhouse of a cell. Golgi apparatus are the storage and packaging organelles. Ribosomes are the protein factories. Endoplasmic reticulum are the organelles for protein, lipids or steroid synthesis. Cells also have small sacs called vacuoles for either storage or waste disposal. Here you can see the differences and similarities between prokaryotic cells and eukaryotic cells. Now let's talk about the animal and the plant cells, how they are similar and how they are different from each other. This is a typical animal cell and this is a plant cell. A plant cell is more regular in shape than an animal cell. A cell membrane is a thin barrier that surrounds every single living cell. It is also known as the plasma membrane or cytoplasmic membrane. Every cell has a jelly-like substance that fills the interior. It is called cytoplasm. It provides a medium for various cellular processes and metabolic reactions to occur. The nucleus often referred to as the control center of the cell, is a membrane-bound organelle found in both plant and animal cells. In animal cells, the nucleus is placed in the center, but in plant cells, it is shifted towards one side of the cell due to the presence of a large vacuole in the middle. These tiny organelles are known as ribosomes. These are responsible for protein synthesis. Animal cells tend to have more ribosomes compared to plant cells because animal cells often have higher protein production needs. Next is Golgi apparatus. Both animal and plant cells have Golgi apparatus or Golgi bodies. In the plant cells, complex polysaccharides of the cell wall are synthesized in the Golgi apparatus, though their main function is packaging and secretion of proteins. It receives proteins from endoplasmic reticulum. It packages it into membrane-bound vesicles, which are often then transported to various destinations. An actively growing cell has more Golgi apparatus. The Golgi bodies are smaller in size, more in number, and dispersed throughout the cytoplasm in plant cells as compared to the animal cells. This is because in plants they perform diverse functions beyond just secretions. Next is endoplasmic reticulum. These are present in both animal and plant cells and are involved in protein and phospholipid synthesis. Next is mitochondria. These organelles are called the powerhouse of a cell because it is inside this organelle sugar is broken down to release energy by the process of aerobic respiration. Cells use this energy for different types of activities. While both plant and animal cells have mitochondria, an animal cell has an average of 1000 to 2000 mitochondria, while a plant cell has an average of 300 to 450 mitochondria. The difference in the number of mitochondria is because the energy demands of animals are more as compared to the plants. Now the next three features are prominent or only present in the plant cells. Number one is presence of a large vacuole. In plant cells, the vacuole can occupy a large portion of the cell's volume, storing essential nutrients like sugars, amino acids, minerals, waste products, and water. The water stored in the central vacuole helps to maintain an internal pressure that helps plants maintain their shape and rigidity so that various cellular processes function properly. A plant shows wilting when the water is lost from the vacuole. Plant cells typically have a single large vacuole, whereas animal cells may have multiple smaller vacuoles called vesicles, which may contain food or water. Next are chloroplasts. These are green colored organelles. Because of this green pigment, the chloroplasts are capable of producing glucose and oxygen by the process of photosynthesis. Animal cells lack chloroplasts, so they have to depend on either plants or other animals for their energy needs. Next is cell wall. Plants have a cell wall and animal cells lack cell wall. This is particularly important for plant cells which can take in a lot of water and swell. Without a cell wall, they would burst. The cell wall provides a strong, rigid outer layer that gives the cell its shape and helps it resist internal pressure. This chart shows the similarities and differences between animal cells, plant cells, fungi cells, and protist cells. We can see that cell wall is absent in animal cells. It is present in plant cells and is present in some fungi and some protist cells also. Chloroplasts are totally absent in animal cells and fungi cells. They are present in plant cells, but they are present in few protist cells. Vacuoles are present in all of them, but a large central vacuole is present only in the plant cells. Mitochondria, endoplasmic reticulum, ribosomes, Golgi apparatus, and nucleus are invariably present in all of them. Cell membrane. A cell membrane is a thin membrane around a cell and is present in all the cells, whether it's a prokaryotic or a eukaryotic cell. It is made up of protein and fat. This thin yet powerful barrier controls what enters and leaves the cell. It is partially permeable, means it allows certain materials to move in and out of the cell and not others, ensuring the survival and proper functioning of the cell. A cell membrane is also called plasma membrane. A cell membrane protects and supports a cell. Plant cells have additional envelope around the cell membrane and it is called cell wall. The cell wall is rigid due to the presence of crisscross network of cellulose. It provides structural strength to the plants. Cytoplasm. 70% of this jelly-like substance is water. Rest are organic molecules such as carbohydrates, proteins and fats. It also has dissolved salts, minerals and other small molecules essential for various cellular processes. Cell organelles such as mitochondria for energy production, ribosomes for protein synthesis, endoplasmic reticulum for protein and lipid synthesis, Golgi apparatus for packaging and distribution, vacuoles for storage, and nucleus, the control center of the cell, are suspended in the cytoplasm. Functions. The cytoplasm plays a vital role in several cellular activities. The cytoplasm provides a medium within which organelles can move and interact with each other. Molecules and materials can move through the cytoplasm, facilitating the transport. Many enzymes essential for cellular metabolism are found within the cytoplasm, enabling various chemical reactions to occur. The signal molecules can travel through the cytoplasm to coordinate cellular responses. Chloroplasts. They are double membrane bound organelles with an internal fluid filled region called the stroma. Suspended within the stroma is a network of flattened membranous sacs called thylakoids. Thylakoids contain chlorophyll, the pigment that gives plants their green color, and plays a crucial role in capturing light energy in the process of photosynthesis. Stacks of thylakoids are called grana. All the green structures in plants, including stems, leaves, and unripened fruits, contain chloroplasts, but most of the photosynthetic activity in plants occurs in the leaves. A vacuole is a membrane-bound cell organelle. It is surrounded by a membrane called tonoplast and is filled with a liquid called cell sap. Cell sap is different from the cytoplasm. Now, what is the function of a vacuole? It maintains the shape of the cell. In plant cells, when the vacuole is full of water, that water exerts pressure on the cell wall. This is called turgor pressure and it helps the plant cell to maintain its shape and keeps the plant upright. It also acts as a storehouse. A vacuole in plants stores carbohydrates, lipids, salts, minerals, pigments, latex, rubber, and sometimes toxic materials too. Flowers and fruits get their colors from the pigments stored in the vacuoles of their cells. Certain plant vacuoles might also play a role in defense mechanism against herbivores by storing toxic materials. In animal cells, vacuoles are generally small and numerous in number. These are called vesicles and they help isolate waste products or toxic materials from the cytoplasm, preventing them from harming rest of the cell. In animal cells, vesicles can help transport materials such as enzymes, hormones that an organism needs to survive and also recycle waste materials. Nucleus. Nucleus is the control center for all cellular activities. It helps the cell to make the right types of proteins. The nucleus houses the cell's genetic material, the DNA. DNA in eukaryotic cells is in linear chromosomes. The nucleus is surrounded by a nuclear envelope that separates the DNA from rest of the cell, safeguarding it from any damage. Nucleus is found in plants, animals, and fungal cells. Bacteria and archaea lack a true nucleus and have their DNA in a single circular chromosome located in the cytoplasm. Mitochondria. These are double membrane bound organelles. Primary function of the mitochondria is respiration, a process of breaking down glucose and other nutrients to release energy in the presence of oxygen. That's why mitochondria are also called the powerhouse of the cell. Animal cells have more mitochondria than the plant cells because of their high energy requirements. Cells of muscles, sperm cells, and nerve cells have more mitochondria due to their high energy demands. Prokaryotes do not have mitochondria. Ribosomes. These are the smallest cell organelles, and they are found both in prokaryotic cells and eukaryotic cells. In prokaryotic cells, they are freely floating in the cytoplasm, but in eukaryotic cells, they are both freely floating in the cytoplasm and attached to the rough endoplasmic reticulum. Ribosomes are the protein factories because in them the amino acids join together to form proteins under the instructions of DNA. Endoplasmic reticulum. These organelles are continuous folds of membranes. There are two types of endoplasmic reticulum. One is smooth endoplasmic reticulum, which is found near the cell membrane. The other is rough endoplasmic reticulum, which is found next to the outer layer of the nuclear membrane. This is called rough endoplasmic reticulum because many small ribosomes are attached to its surface, giving it a rough appearance. Both types of endoplasmic reticulum have different functions. The rough endoplasmic reticulum is mainly involved in protein synthesis, and the smooth is involved with lipids, steroids, and glycogen synthesis. So we see that there are ribosomes which are freely floating in the cytoplasm and some that are attached to the rough endoplasmic reticulum. Both of these have the same function of synthesizing proteins, but they differ in for which purpose they are synthesizing that protein. The protein synthesized by the free-floating ribosomes is used within the cell itself. On the other hand, the protein synthesized by the ribosomes which are attached to the endoplasmic reticulum are transported out of the cell. Now it's your time. Check your progress by answering some questions. The answers are given at the end of the video.